Hello everyone, thank you for being here today. We will having our new topic about proteins. So before anything else, let me introduce myself together with my group mates. I'm Rhea Midegomio.
I am Grinzel Ace Arnado.
I'm Hazen Mecca Allison.
I am Brent Shoshana.
And I'm Craig James P. Asignado.
Okay, so as I mentioned, the first thing I'm going to talk about is proteins. So, what is proteins? Proteins are polymers of amino acids. They serve a wide range of functions including organization, transportation, and defense. It is found in a variety of foods like eggs, dairy, seafood, legumes, meat, nuts, and seeds. The objectives of this experiment is to understand the isolation of different proteins, to explain the theoretical background involved in the different qualitative tests for proteins that have been isolated intact or hydrolyzed, and to compare the denaturation of proteins under different denaturing agents. Our first procedure here is to prepare and gather all materials needed for the experiment 1, gluten from wet flour, and experiment 2, denaturation of proteins. Next is to perform each of the experiments, analyze and observe the results. After that, record the results and answer support questions and make a conclusion. So first of all, I'll talk about the isolation of proteins. Isolation of proteins is the process for separating a type of protein from a complex mixture. Its importance is to characterize the protein solubility, acid-base property, function, structure, and interactions. So for this home experiment, our task here is to isolate the intact protein from wet flour. So let's start with experiment 1, gluten from wet flour. Okay, so here's the materials to be used. First, place the flour into a suitable bowl for kneading. Add enough water and mix until a dough is formed. When the dough was formed, place the dough under running water. Continuous washing was done in order to remove all the starch. To know that the starch has finally removed from wet flour, we undergo iodine test because this test used to detect the presence of starch. By the help of iodine test, we easily detect that the starch and gluten is already separated. Upon adding iodine solution, the dough shows black or violet color, which means there's the presence of unwanted starch. So, we need to wash for some more time. After several washings, we add again iodine solution. And yellow-orange coloration was observed, demonstrating that all starch was expelled. The insoluble material remaining within the dough was recognized as the crude gluten. For visual observations, anyway, we only use one brand of wet flour, which is Happy Panda. So the appearance and texture of our thick dough, its color is white and texture of our thick dough is smooth. As we test the thick dough with starch plus iodine test, the dough shows black or violet color until the iodine made contact with the dough. And then the dough without starch plus iodine test, the dough shows yellow-orange color, and when the dough, after all starch has been removed, the dough becomes gummy and stretchy. After the starch is removed from the dough, it becomes smaller, leaving a slimy network of gluten. Okay, let's have guide questions. First question is, describe the protein composition of gluten. Gluten is made up of two different classes of proteins called gliadin, the water-soluble component, which gives bread the ability to rise during baking, and glutenin, the water-insoluble component, which is responsible for dough's elasticity. The two proteins that bind together will form a network that holds the dough together and makes bread light and fluffy. Next question is, what is the principle involved in the isolation of gluten? The principle involved in the isolation of gluten is a difference in solubility. Starch is partially soluble in water while gluten is insoluble in water. The gluten is isolated by washing the flour with water to remove the starch and the substance, leaving only the gluten. Number 3. How did you assess the complete removal of starch and isolated gluten? Okay, so we tested the complete removal of starch and isolated gluten from wet flour by persistently washing the dough with water to expel the starch from it, and the insoluble material, which is gluten, that isolated from starch. Number 4. Describe what celiac disease is. Celiac disease is an immune disease in which people cannot eat gluten because it will damage their small intestines. People with celiac disease might experience symptoms like diarrhea, bloating, gas, anemia, and growth issues.
The following schematic diagram of the experiment is overview of the isoelectric precipitation of casein from milk using an acid. This method is used to make cottage cheese. However, cream cheese making and the real absorption of casein uses different kind of milk. So what is an isoelectric precipitation or isoelectric point? Isoelectric point is the pH of the solution which the electric charge becomes zero or neutral. Isoelectric precipitation of a protein, when it is at its isoelectric point, it becomes less soluble. In general, the net charge on the protein, either positive or negative, can interact with water molecules, meaning that it's more likely water soluble. As a result, amino acid is the least soluble when the pH of the solution is at its isoelectric point. This does not mean that all amino acids and proteins are insoluble at their isoelectric point, simply that they tend to be less soluble at the pH than at pH values where they carry net charge. So what is the difference between whey from casein? Whey protein has more branched-chain amino acids or BCAAs, while casein contains more of histidine amino acids, methionine, and phenylalanine. Between whey and casein, whey is the faster protein supplement because its amino acids are absorbed quickly by our bodies, while casein is the slower supplement since it's digested more slowly, but both proteins can help our muscles grow. So we're going to go ahead and list the common amino acids that can be found in bean proteins. The common amino acid that is present in bean proteins are the leucine, lysine, and phenylalanine or tyrosine. These are the predominant common amino acids, followed by the valine, isoleucine, and histidine. What are the uses of the proteins or nutrients found in mongo or mung bean? Mung beans help insulin work more effectively and may lower the blood sugar level because these beans are high in fiber, protein, and it contains antioxidants. But if you want to grow your muscles with the help of mung beans, you should pair it with rice to create a complete protein profile. Even though you get lots of protein from beans, it isn't complete. Beans are lacking in one or more of the essential amino acids you need from your diet. If you eat meat, dairy, or seafood, you don't have to worry. These foods already have all the essential amino acids your body requires, which can also help in acquiring complete protein profile. So what is the myoglobin's molecular description, functions, and mechanism of activity? Myoglobin is a protein found located primarily in the striated muscles of vertebrates, and it can also found in bloodstream after muscle injury. It contains a heme prostatic group that can reversibly bind to oxygen. Body uses it as a storage protein in the muscle. It binds and releases oxygen depending to its concentration in the cell. Then this also allows organism to breathe for a longer period of time if it is in high concentration. So the difference between hemoglobin and myoglobin. Myoglobin is a monomeric protein found in the muscle cells, and this uses as a storage site for oxygen in the muscle, while hemoglobin is a heterothermeric oxygen that transport protein found in red blood cell. So what is myoglobin's relationship with rhabdomyolysis and myocardial infarction? Myoglobinuria is usually the result when rhabdomyolysis or muscle destruction happen. It occurs when anything interferes with the storage or use of energy by muscle cells. When myocardial infarction happens, myoglobin is released into the blood, and serum of myoglobin levels may rise. Myocardial infarction happens when one or more areas of the heart muscle don't get enough oxygen. This happens when blood flow to the heart muscle is blocked.
Good day everyone, I am Hazen Mecca Allison and I'm going to discuss the qualitative test for proteins. Qualitative test is composed of two, which is general and specific. Under the general, we have Biuret. Biuret's theoretical background is it is possible to identify the existence of peptide bonds by using the chemical test in this manner. The frequency of a Biuret reaction used to generate peptide bonds is the same for each amino acid in the peptide. Hence, this fact may be used to calculate the protein concentration. The presence of protein is indicated if the experiment becomes purple. Next, under the general is we have ninhydrin. Ninhydrin's theoretical background is this chemical test can be used to determine whether a sample contains aminines or amino acid using the ninhydrin test. The presence of primary or secondary ammonia indicators of aminines or amino acid in the analyte include the emergence of vivid blue color. Ammonia is a amino acid, primary or secondary amines, and deep purple. A yellow color is produced for proline and hydroxyproline. Brown color is obtained for asparagine. The analyte does not change color if no color is noticed, includes amines, ammonia, or amino acid. Next is under the specific is xanthroprotic test. Its theoretical background is a nitration reaction is used in xanthroprotic test to identify the presence of protein in a solution. The sample combines with aromatic amino acids like phenylalanine, tyrosine, and tryptophan to produce a yellow substance known as xanthroprotein when it is exposed to hot, concentrated nitric acid. Next is Milon's test. Milon's test theoretical background is a specialized test to find amino acids is called the Milon's test. Being phenolic or having a hydroxyl group linked to ring, also known as tyrosine, positive findings indicate color break findings that are crimson, which are tyrosine and phenol solution. Displays no red at all, it is anginine. Next is we have this Hopkins-Cole test. Theoretical background about this test is the Hopkins-Cole test is used to identify a protein's tryptophan. For a successful outcome, it displays a purple color. Next is we have this Sakaguchi test. Theoretical background is a chemical test called Sakaguchi test is used to identify proteins' arginine content If it displays a red color, it indicates a positive result. Next is we have lead acetate test. Its theoretical background is when identifying amino acid like cysteine, a biochemical test use the lead acetate test or lead sulfide test. This test is specifically designed to find sulfur-containing amino acid, and the presence of black precipitate indicates the presence of sulfur. This is nitroprusside test. Its theoretical background is this test only detects acetoacetate and acetone, but is used to detect the presence of ketoacids in the blood and urine. Because it cannot detect the presence of beta-hydroxyl butyrate, it may underestimate the severity of ketomania and ketonuria or BOH. The presence of a reddish complex during the nitroprusside test serves as a sign of success or it's positive. Next is the Sullivan's test. As theoretical background is that it is used to determine whether cysteine is present or the protein cysteine. When it's color red, that's why cysteine are after being heated with sodium. 1, 2 negative cysteine is present, sodium and naphthoquinone for sulfate. Methionine when the pH is alkaline. Second to the last is we have Pauli reaction. Pauli reaction's theoretical background is Pauli reaction must be performed to determine whether tyrosine or histidine is present in proteins. It displays red color complex when histidine and tyrosine are present in the solution. Last is we have Lieberman's test. Theoretical background of Lieberman's test is that cholesterol may be found using the Lieberman's test. For a few minutes, if the color changes to green or green-blue, it is a positive indication.
Hi, I'm Wrench, and for now, we will be presenting our second experiment, the denaturation of proteins. Before we start, these are the materials we used. So first, pour each of the solvents in the glass container, then label. For the sunlight, have it remained empty and label the procedure progress to adding of the egg white. Second, crack egg into the bowl, removing the yolks. Third, cut the egg white into pieces so you can add equal amount into each glass container. If possible, to add only at least 10 to 15 milliliter or 1 tablespoon of egg white to at least make it visibly floating in the solvents. Fourth, control glass remains to have egg white alone, while glass for sunlight must be observed under direct sunlight in the same amount of time with the other glass. Fifth, observe any immediate changes that occur in terms of egg white color and consistency. If you try stirring the different treatments, rinse your fork between stirs. 6. Wait for 20 to 30 minutes. And last, use the fork to inspect the state of the egg whites in each treatment and note how they may have changed over time. Thank you. That's all for our experiment number 2. So, based on our visual observations on the egg white being mixed with different denaturing agents, for solvents we have the tap water, detergent, vinegar, alcohols with 45 and 70% solution, and for the heat we will be using the hot water and sunlight. On the other hand, we have the control which no added agents, only the egg white alone. So, from our observations with the tap water, the egg white in the tap water forms a very fine white thread or forms into small coagulated parts. Next is the detergent. The egg white and detergent blended together after being mixed. It expanded and ended up not visible. And from vinegar, the egg white in the vinegar did not coagulate. And from our 45% solution alcohol, the egg white starts to turn cloudy and coagulates into a fair size. While from our 70% alcohol, same result in 45% solution of alcohol, the egg white starts to turn cloudy and coagulates into a fair size, but it forms bigger and in many parts. And from hot water, the proteins unfolded, taking up more space, instantly coagulated, turning the gel white, and hardening them in place next to one another. Next is the sunlight. So, from our sunlight on the day of our experiment, there wasn't enough sunlight on our 30 minutes basis. That's why the white color is not really visible, but we must take note that the egg white under direct sunlight for 30 minutes slowly started to coagulate and turn to color white, for it only depends on the heat of the sunlight gives. So that's all for our visual observations. Let's move to our questions. Question number 1. Describe the mechanism of action of the denaturing agents used. In detergents, it affects R group interaction. And for the strong acid, it disrupts hydrogen bonds and salt bridges. Prolonged action leads to actual hydrolysis is peptide bonds. And in alcohol, it interferes with R group interactions because these solvents also can form hydrogen bonds. Quickly denatures proteins in bacteria, killing them. For example, the disinfectant action of 70% ethanol. And for the sunlight, it operates very similarly to the action of heat. Example, sun burning, not the midnight sun. So let's move to question number 2. What are cheotropic agents? A cheotropic agent is a molecule in water solution that can disrupt the hydrogen bonding network between water molecules and reduce the stability of the native state of proteins by weakening the hydrophobic effect. Question number 3. State the negative effects of denaturation in our system or body. Serious eye damage can result from eye tissue contact with acids of bases. When irreversibly denatured and coagulated, protein causes a cloudy cornea. Question number 4. Describe the other major proteins in albumin. The liver produces albumin, which serves to regulate the osmotic pressure of the blood compartment, nourish the tissues, and transport hormones, vitamins, medicines, and other chemicals such as calcium throughout the body. Question number 5. What are the functions of albumin? Albumin is a water-soluble protein found in egg white as well as milk and blood serum. It refers only to a specific class of proteins coagulated by heat which are found in the interstitial fluids of animals. Substances contained with albumin are called albuminoids. Albumins dissolve in water and solidify into a semi-solid substance when heated, which explains why egg whites become solid and white when cooked. Question number 6. State the relationship of albumin with the protein energy malnutrition, marasmus, and kwashiorkor. Kwashiorkor causes nutritional edema and metabolic abnormalities, such as hypoalbuminemia and hepatic steatosis. In children, whereas marasmus causes severe wasting. According to studies, marasmus is an adaptive response in starvation, while kwashiorkor is a maladaptive response to starvation. Protein deficiency causes rapid loss of cellular ribonucleic acid and disaggregation of endoplasmic reticulum-bound polysomes, resulting in reduced albumin synthesis. Question number 7. Explain why we are using 70% alcohol as better disinfectant and not 95% absolute alcohol. What is 45% used for? So, why? Because they dry out in seconds, whereas the average contact time for most disinfecting agents is at least a minute. This means bacteria or germs aren't contacting the disinfectant long enough for them to be killed. For this reason, to achieve the appropriate contact time, IPA is diluted and watered to around 60 to 70% so that it evaporates much slower. For instance, 70% of alcohol solution contain about 30% purified water. This water content plays a crucial role in inhibiting pathogenic microorganism growth. So, for the last question, differentiate 70% ethyl alcohol from 70% isopropyl alcohol. The most common hand sanitizers are isopropyl alcohol, aka isopropanol, 2-propanol, or just rubbing alcohol. You cannot drink this stuff without getting sick, and it will not get you drunk. The alcohol you can drink is ethanol, aka ethyl alcohol or grain alcohol, since it's the kind of alcohol in any liquor that distilled from grain. You can get drunk on this stuff, your tolerance may vary. The ethanol in hand sanitizer, however, is denatured and will not make you drunk, but it will make you very sick. These two alcohols have similar structures, but then chemical variations are enough to make one drinkable and one dangerous to ingest. But when it comes to hand sanitizer, they work the same way. They both disrupt the proteins and lipids in viruses and bacteria, which kills those germs. So that's all. Thank you.
So good day everyone, this is Crinzel Ace Arnado speaking, and now I am going to conclude our reporting based on the information we have gathered. So we conclude that proteins are the most elaborate of life's molecules. A protein is a polymer constructed from amino acid monomers. Every human body has tens of thousands of different kinds of proteins, and each protein has a unique three-dimensional structure that corresponds to a specific function. Proteins perform most of the tasks the body needs to function. They can be classified based on their function, and these may include structural. Proteins also play an important role in making chemical reaction in the body occur faster. Without proteins in the cells, chemical reaction would be too slow to maintain life. The basics that have led to the understanding of these have led to the understanding of the mechanisms behind the isolation and denaturation of proteins. Through the experimental procedure, a better understanding of proteins has been obtained. And for our nursing application, studying proteins is very important to healthcare professionals in order to identify their functions and to understand how the genes that produce them are altered in disease. As a student nurse, it is crucial to understand how proteins work in our bodies in order to know the factors that affect how much protein a person consumes within a day, and the underlying condition if a person has excessive or inadequate protein in the body, and also to understand the importance of nutrition basics to be able to explain the facts about healthy food choices to patients. As a healthcare professionals, we already know that proper nutrition is not only important for preventing disease, it is also crucial to recovery process of the patients. Furthermore, due to the importance and varied roles that protein has in the body, it is vital in assessing the movement of nutrients in and out of cells essential to building bones and body tissues, and most especially a nutrient in wound healing. For instance, protein deficiency is widely held to be a major problem in malnourished patients and has been linked to an increased risk of pressure ulcers and slow wound healing in a number of studies. However, maintaining a proper dietary concentration of protein is crucial because if someone consumes too much or too little, they run the risk of developing health problems. So if we can understand the function and precise mechanism of protein causing a disease, we could design a drug to relieve the symptoms. So that brings us to the end of our presentation. And at this point, we would like to know if you have questions, qualifications, violent reactions, addendum, or concerns with regards to our topic. And if none, we're looking forward to know that you've learned our topic today. And thank you for listening, and may God bless us always. Padayon future nurses.